Just a quick video on how to run the in silico activity that is going to take the place of class today, if you're watching it that day. First of all, go to our Blackboard site and click on the Hydropathy plot in silico activity folder, which will open up this page. There are directions, step-by-step directions, so I'm going to open that up. Okay, please be aware that there are step-by-step directions for you. I'm not going to keep flipping back and forth between these directions and what I'm doing. I'm going to start with task 1, step 1, and just kind of move through that. If you click on these links, they will open up the pages. Okay. Also, you want to download the activity sheet, which is here. So I'm going to do that and open it up. And that will open up the activity sheet that's going to be due for that day's class. There is also in this area a protein for hydropathy plot activity. Each one of you is assigned one. I have all of them in here, but I'm going to open up my instructor one. And so I have 2 of them that we're going to work through. You will only have 1. I just want to show you both a hydropathy plot for an integral membrane protein and a hydropathy plot for a peripheral membrane protein, because there's a mix of them going on. So I will start in the directions. First of all, we want some very specific objectives. We want to be able to generate a hydropathy plot, run a blast search, figure out what the actual protein might be, and then use the hydropathy plot to support the blast search identification of that protein. Remember that hydropathy plots look something like this, and if you read this background, you're going to see a hydropathy plot on the left for an integral membrane protein and one for a peripheral membrane protein, and we're going to be using freeware. I do want to put out a shout out. I know I'm talking fast. I just want to get you through this, but the Swiss Institute of Bioinformatics, SIB, is one of the biggest websites for, biggest institutions for genomics, especially for protein analysis. They have an expert protein analysis system called XPath that is really wonderful. And the US is leading the game as well with the National Institutes of Health, NCBI, this basic local alignment search tool, which is called BLAST, in case you already don't know that. Kudos to them for making this free software. And I'm just going to run us through all these steps. So first thing I am going to do is I am going to get my entire sequence here. So I'm going to go from M, which is methionine, all the way to the end of the protein sequence, and I am going to copy it. I am then going to open up my hydropathy plot XPath software, and I will paste in this entire sequence from methionine all the way down to A. The next thing I will do is I will go to the Windows size here and set it to 19, which makes sense for what needs to get through a membrane, and I will hit submit. And I will get a hydropathy plot for this particular protein's amino acids. I am going to go into my activity sheet, and because it's already still in here, I can paste my amino acid sequence here. Okay. I'm also going to open up my next site because I already have my sequence copied and I know I need to do these 2 tasks with it. So I'm going to find BLAST and I will paste my entire sequence into there. Hit 9606 in this area here for Homo sapiens. And then I'm going to hit blast search. Okay, so I've done both my tasks. I've made a hydropathy plot. I've got my blast search going. I've opened up my sheet and I've put in the first 6 letters of my particular protein. I'm going to right click on here and I'm going to copy this image and I'm going to go back to my activity sheet and I am going to paste it in question number 2. I'm going to grab my draw here, and very importantly, I am going to draw an arbitrary line at 1 here. There's a lot of noise in a hydropathy plot, and it's pretty important to kind of cut out this background noise between 0 and 1 and find a true peak. So 1 definite transmembrane domain, 1 putative transmembrane domain. This protein has 1 or 2 transmembrane domain, and I am going to predict that it is an integral membrane protein. So I am going to go to my blast search, and I am going to find out that based on that amino acid sequence, this profile is telling me that it is a signal recognition particle receptor. Okay, so that is what it's telling me. I'm going to copy that. I'm going to go back to my sheet, and I am going to undo that because I pasted it in the wrong place. Paste it here because it's telling me what you find on your blast. Now this is the hard part. You can Google this, or you can go through our notes and find out what these things are. But I've chosen nothing that we haven't seen in lecture before. This is an SRP receptor. Where in the cell is it located? In the ER. Is it integral or peripheral? It is an integral membrane protein embedded in the ER membrane. Good. And now I'm going to use that information to explain why this hydropathy plot supports this being a blast. SRP receptors are integral membrane proteins, and as such should have at least 1 transmembrane domain. The hydropathy plot shows a protein that has 1 or 2 transmembrane domains, and as such supports the blast search results. Okay. So I am done with my worksheet. I'm going to save it. So I am going to save as, I'm going to export it. I'm going to export it as a PDF onto my desktop, and I'm going to be like AJL hydropathy plot, and I'll publish that here. And now I'm going to go back to my Blackboard site, and I am going to go into our online assignments in this folder submission, find the In Silico daily activity. I can view the rubric if need be, and then I'm going to browse my local files, find my desktop, AJH plot, hit open, and I am going to submit that. Okay. And I'm done for the day. Good. If you want to stick around, I'm going to show you the opposite. I'm going to go back to my beginning, and I am going to show you what a peripheral membrane protein looks like. So I'm going to reset that. I am going to go back and get another one of these. I'm going to cancel that. I am going to get a fresh copy of that worksheet just quickly so that we can look at it together fresh. I'm going to get that worksheet from here, and we can start again. Okay, so I have another worksheet. I have another one. You will not have another one. You will only have one. I just want to show you what the flip side of this looks like. So I'm grabbing another amino acid. I'm going to stick it in here. I'm going to set that to 19, and I am going to submit that. I am going to go to my blast. Good. I'm just going to clear this old one here and put my sequence in here, make sure I have 9606, and hit blast and let that run in the background. I'm going to go to my fresh sheet here. I'm going to close that one, or else I'm going to keep opening it. I'm going to go to my fresh sheet. I'm going to paste my sequence in there because I'm done with my sequence now. I'm going to go back and capture my hit submit and get my hydropathy plot. You can appreciate that this one already looks different. I'm going to copy that and put it into my sheet. Feel free to leave the video if you kind of know where you're going. I am going to get my draw function here, and I am going to draw a line at 1. Now my scale doesn't have a 1, so I'm going to be like, it's up here somewhere. Okay, already I know this is a peripheral membrane protein. There are no transmembrane domain peaks here. And I'm going to go back to my blast search, and I am going to find out that it is cytochrome C according to the blast search. So I'm going to go back here. I'm going to say my blast picked out cytochrome C. I keep putting it in the wrong place, but that's okay. You guys will do better than me. Where in the cell? You can Google it. It's in the mitochondria. Is it a protein that is integral or peripheral? It is a peripheral membrane protein in the intermembrane space of the mitochondria. It's part of the electron transport chain. And then I'm going to use this information to say cytochrome C is, okay, I'm just going to say it is, because I can't type for now, peripheral membrane protein. And as such should have no, that would be 0, transmembrane domains. The generated hydropathy plot shows a protein with no transmembrane domains, and therefore, I'll just put and therefore supports the blast search, something like that. I'm going to save that. Actually, I'm going to export it as a PDF onto my desktop, and I'll keep it like that, and I'll publish it, and I will go back to my Wiley Plus and other materials, my online assignments, scroll down, find that in Silico daily activity. I will browse my local files, find it on my desktop, find the right one here, and open it. It should get attached, and I will submit it. And I am done. So hopefully you have fun with this, and I will see you next time on the flip side.